Welcome, everyone. My name is Jackie Barendregt from Pharma. I'm Chief Operating Officer, and welcome to our webinar on how to enhance biopharma's path to market. We have a lot of interest in this topic from our leaders around in the biotech industry, working globally from clinical development, regulatory, as well as commercial. So we'll make sure to be covering all of these different areas in today's webinar. And today I'm joined by two of my colleagues here who will introduce themselves. Alex, please.
Yes, certainly. I'm Alex Graham, the Chief Medical Officer of Biopharma, and as the title suggests, obviously my focus in the organisation is around the clinical development components of any path to market. Aiden, over to you.
Thanks, Alex. Hello, everyone. So I'm Aiden Flynn. I'm the founder and CEO of Exploristics. I've been working in statistics for 30 years now. I've always had an interest in how data and statistics and modeling can better inform decisions throughout clinical development.
Thank you. And what we'll be covering in today's webinar is a quick overview in terms of our philosophy and approach, how we think about path to market design, as well as how we can improve decision making with evidence-based decision inputs. And then we'll actually provide you with a case study demo that we've put together specifically for today's session. And after that, we're happy to take any questions, get your experience in terms of bringing molecules to market successfully, or also some of the challenges and the barriers that need to be overcome. Feel free to drop any questions in the chat so we can pick these up along the way or towards the end of the webinar. So starting with a quick overview in terms of our philosophy and approach, because we're two companies, Exploristics as well as Biopharma, with a shared philosophy around improving evidence-based decision-making. So we can, you could say, enable more higher quality in terms of these decisions, but also kind of shorten the time from molecules in the lab into the market. And our main approach, as I mentioned, is helping companies make better decisions. And this really starts in early phase. So we develop product launch and lifecycle management options and put that into a strategic decision-making framework. And the path to market for a medicine is never obvious. So we need options that meet every hurdle, clinical, regulatory, and the most important commercial hurdles. Clearing any one of these hurdles can be relatively easy, but clearing all of them is complicated. And 1,000 decisions really determine the trajectory of a product. So products are ultimately the result of what you've learned and decided where you take your molecule from the lab into the market. And that's why we need to change early phase development from the prediction paradigm to asymmetric learning, from predict and confirm to explore and execute. Aiden, can you elaborate a bit further on how you support this with Exploristics?
Yeah, sure. So we've built a clinical trial simulation platform called Keras Cloud. And what that does is allow us to evaluate the performance of different study design options or different development pathways in silico before executing the actual plan. So we can generate lots of virtual studies and pathways and evaluate their performance in terms of probability of success and duration. And really the way that we work together with IDEA is looking at those decision trees that that summarize the different development pathways, and we provide the inputs in terms of probability of success and duration into those decision trees.
Great. Thank you, Aiden. In terms of our collaboration also with global biopharma clients is that we help clients develop these different options in terms of launch and lifecycle management for their assets as well as portfolios. And this really evolves around thinking about your products and your molecules as not being average and thinking about the opportunity that sits within the market on one side in terms of customer goals, unmet medical needs, as well as then you could say the opportunity from a scientific and biological point of view that your molecule might bring, and really bringing that all together. So we work with clinical, regulatory and commercial consultants across a range of therapy areas, across a range also of different therapeutic modalities, ranging from small molecules to biologics and cell and gene therapies. In terms of our philosophy and mentioning around, you could say, the decision-making framework, we have developed a methodology called 5X. And it's a novel path to market discipline that really pushes teams within the pharma companies to innovate. And it was built on the longest study of pharmaceutical innovation ever run. For people who might be familiar with our Pharmaceutical Innovation and Invention Index. And the main lesson of this study is that molecules do not self-determine their opportunity. It's really the people who work with those molecules. And this may sound obvious, but we see again and again, you could say, case studies within pharma where you have almost similar molecules or even similar molecules ending up in very different places in terms of the commercial potential that they've been able to realize. So now this principle around, you could say, innovation for us really means, are you able to generate also return on your invention? Because sometimes there's a lot of focus on the invention itself rather than really realizing the value that it could bring into the market. So now the goal of path to market strategy is to evaluate where your asset can go and what the market might want from your product, from your asset. And these two goals are clearly interdependent, as I mentioned at the beginning. So competitive advantage will come from teams who gain insight into that more quickly than other companies. In terms of, you could say, developing these different options, we enable teams to think about strategy in, you could say, a creative way. And what teams need to bring is, you could say, the highest levels of knowledge and creativity, and an approach to gaining insights quickly. So in terms of our 5X platform, it creates a range of options where you would have an asset with the fastest path to market. You would have an index patient approach, which is typically the precision medicine approach, a lowest cost path to market, the expected path, what would any other given company do with a similar molecule and capabilities, but also what would be the unexpected out-of-the-box path to market. And it allows to, when you think about, you could say, these different options of where you could end up in the market and how you can travel from A to B with what are the risks on the journey, as well as the opportunities for success. And this allows to compare and contrast these options and ultimately give leadership and decision makers the opportunity to diversify their portfolio for gaining competitive advantage. In terms of how we then support teams with decision making, Aiden, do you want to pick this up here in terms of optimizing the design?
Yeah, sure. So I mentioned Keras Cloud in my my intro. So and the way it works is, we build virtual patient populations, so a large synthetic data set, and then mimic the way a clinical trial would work by asking lots of what if questions. What if we focus on a particular group of patients? What if we choose one endpoint versus another? What if we change the duration? There's a kind of an almost unlimited number of what if kind of scenarios that you can evaluate with Keras Cloud. But it forces people to really go out and gather together whatever information they can about the indication of interest. So it builds all of those different data sources and knowledge base into a set of simulations. And this is something that can be updated all the time as new information becomes available. But what we know now is that this approach makes a big difference. We've published papers around, being able to increase the chance of success for a single study by 41%. If you look at an entire development pathway, increasing the likelihood of success more than threefold. saving $20 million on a single study, just by allowing the sponsor to look at a different endpoint rather than the one that they had originally chosen. And we've shown that this can drastically reduce the duration for an entire development pathway as well.
Excellent. So what I'm going to do is I'm going to take you through how we do this right from the start to the end game of looking at these different options. And we're going to do this through the medium of a case study, because it's always much better to, you know, show you something real rather than just the theoretics behind it. Now, the important thing with all of these is it has to start with the science. So if you kind of go to the next slide, we've chosen this particular example of blocking the interaction between CD40 and CD40 ligand. Some people may be familiar with that, but it's an immune mechanism. It's one that's been looked at in a number of different auto-inflammatory conditions. I'm sure if you're familiar with that space, you'll have heard about it. And this is an example, and this is very common in modern therapeutics, where we've got a target that could have potential utility across quite a wide range of different disease states. And then within those disease states, there may be also segments of those diseases, different phenotypes, genotypes of patients, where there may be more value in pursuing, you know, blockade of the particular target. So the way we operate is we start exactly from the science. We understand what possibilities could exist. You can see on the left-hand side of this slide, this is a map showing some different conditions. This has been produced actually from an AI engine called Causally that looks at associations, biological associations. If you modulate different components of certain pathways, for example, what are the biological consequences of doing that? And then the end game, of course, is which particular disease states that modulation may be important in. So we use that kind of approach. Obviously, we dig into the literature. There may be other inputs from clients that we're working with, or specialists in the space, or scientists that we work alongside who also can provide input. But the aim here is to generate a wide list of potential diseases and segments within them that we could go at. And this is an example of this, where you can see out here listed actually a very wide range of different conditions where that CD40-CD40L interaction and breaking that interaction could have potential therapeutic value. There's actually a much longer list than this even that one could pursue. And you can see on the top, we've just marked in very basic principles where there's stronger and weaker evidence. Causally helps us to dissect that out. You can look at the degrees of evidence that sit behind inhibition of this particular interaction in different disease states and patient populations. And in addition to that, we look at biological plausibility ourselves. We have ways of ranking that so that we understand that we are starting from a good, robust scientific basis before we start to make any decisions going forwards. Now, on the next slide is just a reminder, you know, this matters an awful lot, because if you only look at a narrow spectrum of potential opportunities in terms of either the disease states or places within those disease states that you could operate, that you've really defaulted. And of course, sometimes you have to default into certain areas because of the work that the company has been doing preclinically. But what's also useful about this exercise is it can feed back in, and sometimes some of our clients have chosen to do more preclinical work as a result of this kind of approach and the analytics that we bring forwards, because they do want to generate a wider range of potential preclinical evidence. But this is all about optionality at this point, different diseases and different populations within them that we could go at. So if we go down to the next slide, this is just an overall schematic of the way that we're operating. You can see that generation of ideas and hypotheses sits at the left-hand end. That evidence that's accrued behind those particular disease states and the mechanism involved. The biological plausibilities I mentioned that we can even measure. We have tools to get at that. And the next phase of the process is then to generate what we call target opportunity profiles. And this is very much like generating a broad range of TPPs without the same levels of depth as one might expect in a normal TPP. But it's starting to look at the end game of if we were going to go forwards, what would the indication look like, and get to some specificity around that. So it's an alignment, obviously, between the disease state, the patient population, the kind of outcomes that could be of interest. And of course, there's plenty of opportunity when we look at those opportunity profiles to consider different alignment between those things I've mentioned, as well as things like different durations of the trials that could be run, different comparators that could be inserted, some of the things that Aiden has already alluded to. So even for a specific disorder and a specific patient population, there may be multiple ways of considering what that target opportunity profile could look like. The important thing from going from the, if you like, the theoretics of the biology into this harder edged profile is it does exactly that. It puts edges around it. It raises questions of all of those things around the patient population, the endpoints, durations, comparators, etc. Allows us to play around with those and consider what good could look like and generate a range of plausible candidates. It also then, of course, allows us to start measuring things, and we'll come on to that pressure testing and the risk-reward profiles in a moment. But obviously, if one wants to do good analytics, you can't do that on an idea, a theoretic concept. We want to be able to measure things accurately and look at those different parameters that I've just outlined. So Aiden will come and talk about his component of that from a clinical perspective and looking at different ways that we could run trials and manipulate those variables to enhance our chances of probability of technical success. We work as a multidisciplinary organisation, not just looking at those clinical components, but as Jackie already alluded to, looking at regulatory and commercial as well. So we ask questions around, is there an existing pathway and route through from a regulatory perspective? What precedent might exist? Is this novel? And then we can look at the risks associated with going away from precedent, for example, and trying something different with the regulator versus going down more staid approaches that have already been conducted. And from a commercial opportunity perspective, we can then map out the scale of opportunity from these different opportunity profiles, and we can consider the risk and rewards. There may be other issues like barriers in the market, stronger competitive sets in certain areas versus others, for example, and that again can all be very rationally mapped out. So the process overall goes from that generation based off the biology through production of target opportunity profiles covering a range of different disorders, different populations within them, and then those variables of endpoints, etc., that I've outlined, to really thoroughly pressure testing those different options and providing clinical, commercial, and regulatory metrics by which you can judge what good looks like. Is there a good balance of risk rewards in different areas? How do they differ from each other? And back to Jackie's comment also, do we want quicker? Do we want bigger at the other end? What kind of profile are we interested in? We're obviously generating a range of them, and they may over-index in one of those areas versus another, and we can analytically look at all of that and make sure we understand the pros and cons. So I'll just continue to take you through the process that we did for the CD40L. I've talked here about TOPs. This is really important. It also allows you to map back from what you're going to require in phase 3 and start to ask the questions of what you'd need to do in mid-phase developments. If you're sitting in phase 1 and you're just doing an oncologic and you're doing safety, this is absolutely the right time to do this, because it's going to instruct different approaches to how you could conduct phase 2. We all know once you've sat in mid-phase development, it's unlikely you're going to substantively change course between phase 2 and 3. So that's the optimal time to consider this. And if you're doing it in oncology, we'd even advise you do that earlier, maybe as you're starting to dose escalate, for example, but preferably even at the end of the preclinical piece, because you really need to get your ducks in a row before you start embarking on your efficacy work in early phase in oncology. So if we go down to the next slide, I'm just going to put up a demonstration here. We actually have a system where we can log all of what I've just described to you in an online platform. And this means that not only us at our ends in the consultancy, but obviously people within a biotech or a pharma company can have a repository of all of the stuff that's being generated. They can add, change, delete all of this and play around with it. They can do it in different time zones and geographic locations. It means it can all be done offline. Obviously, we have meetings with the team, that's for sure, but this is a way of keeping on generating ideas. And I think very importantly, iterating them, pressure testing them, saying, yep, that's a great idea, but we need to change the patient population a bit there, and just having that flow through of ideas and cross interaction between commercial, regulatory, clinical, etc. in the mix to help and not just generate, but develop the ideas. So we're going to just show you an example here. We actually picked on something that's slightly more off-piste for CD40, CD40L, which is to go down the route of Alzheimer's disease. This is obviously looking at the neuroinflammatory component that's being unraveled, still quite poorly understood, but obviously inflammation is a target in Alzheimer's disease. And you can just see here an example of laying out what this opportunity profile looks like. So there's details in here about the nature of the patient population that we would be getting at. And you can see all of the detail that you would provide in here about what the phase 3 would look like, what the phase 2 would look like in terms of structure, comparators, endpoints. So you can see here, for example, looking at listing out some of the secondary endpoints that we could consider. And you can see from this that you can vary lots of things within here. So you can have multiple profiles that you could look at, varying populations and endpoints, for example. And Jackie's just showing you another one here where we've looked at, ALS was another one we looked at. There's relapsed multiple sclerosis as well. So obviously you can look within a disease state and have multiple opportunity profiles, or across some different disease states, or indeed a combination of those two things, which is exactly what we've done in here. I mentioned that you can go through and we can rate various things. So this is an example of looking at biological plausibility. You can write down all the reasons why it's graduated in that way. We have a scale. It's not just thumb in the air for moderate, by the way. There are clear criteria for why you would give it such a ranking. And all of this information can be stored in here, as I say, and shared with the team and debated and modified, and draw on the expertise of the multidisciplinary team from the company working on the particular product. We can also actually, Jackie, if you just go to the prioritized ones, that would be great. You can then see this overview where it looks across the different parameters. So you can see technical, regulatory, the scale of opportunity, the kind of investment required. That, by the way, numeric figures of the scale of investment needed for the clinical development programs that have been outlined in the materials I've just shown you. The numbers piece is looking at the kind of risks that are in the market that we may be up against, the probability of commercial success. And then you can also see the time to market component. Obviously, we can also map out how long we believe it would take to run certain clinical programs, the timing between those phases, the time with the regulator for submission and approval, map those timelines out, and that again can be added here. So what this is doing is providing a dashboard across this range of different clinical and commercial and regulatory parameters, and it allows you to compare and contrast them. And we can also assign the 5x piece as well. Is this, an unexpected pathway? Is this a particular index pathway? And we can assign that to it. So again, we maintain this range, and we can look at the pros and cons of each of those across the different disciplines. So we have a balanced view. We all know that it's very hard to get something that's clinically going to be very quick, have very low technical risk, be easy with the regulator, and make lots of money at the other end. That's not easy. They usually have got ups and downs like a graphic equalizer. And this allows us to look at all of that and understand the pros and cons across all of those disciplines, not focused on any particular discipline. So it's very much a process of running right from the science through to the way we could potentially operate at a label level, doing the working out of what we believe it would take to get from early phase through the different phases of development, doing the analytics on those 3 core discipline areas, and then being able to weigh up the pros and cons. Now, obviously, as I mentioned in here, we're going to come out with different approaches, either because there are different disorders or different ways within them that we could conduct our clinical trials programs, different endpoints, populations, as I alluded to. And this is where the work that Aiden and Exploristics do comes into the mix, because we really need to understand what's the impact of making all those different variations? How does it impact the scale of the trials? And particularly, of course, the chances of success that we may have. So the probability of technical success if we ran the tape differently in our mid and late phase trial programs. And Aiden, if that's okay, I'll hand over to you at this point. You can talk a bit about our case study here, where we're talking in the Alzheimer's space, and in this case, in mild cognitive impairment. That's the segment of the disorder where the patients have amyloid, they have a degree of cognitive impairment, but they don't have full-scale dementia. So these are patients with cognitive impairment, but no substantial functional impairment. And Aiden and his team looked at a number of different ways that we could conduct trials in that space to map out those variables I've just outlined. So Aiden, over to you.
Yep. Thanks, Alex. So yeah, I'm going to build on this Alzheimer's disease MCI case study, looking at CD40 as a target. This is a hypothetical scenario, really evaluating the potential effects of treatment in terms of progress or progression from MCI to Alzheimer's. The results presented are really for illustration only. One of the big questions that we get asked is, well, where do you get your data from to run the simulations? And we have built some tools which will go out to the literature and extract the statistics from unstructured and structured data sources, and then those tools will kind of process, align, clean all those statistics to give us some kind of synthesis or synthesized view of what's out there in the literature. If you look at the plot at the bottom left, which is just a summary of ADAS-Cog from lots of different trials, you can kind of see the extent of the variability there. And to me, this really illustrates one of the big problems within designing clinical trials, because the truth is you can go out to the literature and find a paper and use that paper as your underlying assumptions for the study design. And you can use different papers to justify virtually any study design. So a key part to this is to make sure that we're using data and sources that are more representative of the study that you're about to embark on. What's also useful in doing this exercise is you can find information from other indications as well, which is sometimes relevant and can be used as part of the simulations. For example, biomarkers or changes in biomarkers as a result of treatment. So in this example, really what we're going to look at is a time to progression endpoint using a clinical dementia rating scale, and also the Alzheimer's disease assessment scale, the ADAS-Cog endpoint as well. So now I'm going to attempt to share. And can you see my screen?
All good. Yes. Thanks.
Okay, good. So this is Keras Cloud. This is the dashboard that you might see if you log into the platform. I'm going to go straight into a project which is relating to this case study. So the way, as I mentioned, the way that it works is you build a virtual patient population first, and that comprises all of the characteristics that are important in the disease, all of the outcomes of interest, the patient level risk factors of interest. Is there any missingness or whatever? And typically we would actually build multiple different populations to just check that a study remains robust against some factors that are perhaps out of your control. You don't always know how effective the drug is, for example. But once you have that virtual population, then you can run lots of different design options. So How do you sample from the population? How many samples do you need? Do you sample all at once? Or do you take a sample, learn from the results and adapt the study? What's the best way to do the analysis of the data? How do you turn that analysis into a set of decision criteria whereby you define success? So that's the kind of workflow that we go through. There's quite a lot of info in here, so I'll just quickly kind of take you through each part. The first part is defining those scenarios. In this case, we're looking at 3 different scenarios where the drug doesn't work, where it has a kind of medium effect, and one where it has a good treatment effect. Then you go in and you use the data from the literature to start putting some numbers against those endpoints. I'll just take you into the ADAS-Cog one to give you an idea as to what this looks like. You put in the averages for the different scenarios and also the likely standard deviation for those scenarios as well. Okay. So let's move on. What you can do as well is link variables to each other. So there's a correlation structure between variables. So in this case, ADAS-Cog is related to the time to progression. I'm not going to go through the other parts because, oh, sorry. Not going to go through the other parts because it's more complex. You can add in missingness and strategies for handling that missingness. Going to the study design approach, all we're doing here is looking at, well, what if we run a study on 300 patients or 400 or whatever? How are patients allocated to different treatment groups? In this case, it's an equal allocation. How do we choose to analyze the data once we get it? And in the simple case, what we're doing here is just a linear regression, looking at ADAS-Cog as the primary endpoint and how treatment and some baseline factor for CD40 and an interaction effect as treatment. But you can build these statistical models to do the analysis. You can have different designs. In this one, we're just looking at a fixed design, but you could have adaptive designs where you're stopping at some interim point and having a look at the data and then adapting this study. And finally, you do all of that analysis and then you need to turn it into success criteria. So a simple one could be, we're looking at that ADAS-Cog analysis, and we're simply saying success is based on achieving a p-value of less than 5%. And we're looking at, different time points as well. So, 6 months, 12 months, 18 months as well. And you can build all of those into the analysis and also the success criteria. You can build more complicated definitions of success whereby you're combining different features, such as you want to have an effective drug, but you also want to make sure it's safe. So you can build in safety and efficacy at the same time. So we're looking at a lot of scenarios in here, different endpoints, different designs, different effect sizes, different durations of the study as well. So we need to sort of summarize all of that somehow. So for each scenario, we're running thousands of virtual studies and working out which of those have met the success criteria. And what you get is a heat map like this. The first segment of this is the scenario where there is no effect. And just to orientate you a bit, across the top, we have all of the different design options. Vertically, we've got all of the different scenarios, endpoints, duration of study, that sort of stuff. Blue indicates that the likelihood of success is very low. And as it becomes more orange and red, the likelihood of success increases. You can see for this first segment that where there's no effect, the chance of success is very low, which is exactly what you want. If we move down to where there's a kind of medium effect, still the chances of success increases, but it's still not where you would want it to be in order to really progress with a study like this. Oh, if we move into the larger effect, you start to see some kind of interesting things. One is as you increase the size of the study, not surprisingly, the likelihood of success also increases. What you can see as well as you increase the duration of the study, the chance of success increases, and this varies quite substantially as well. Also looking at different endpoints, they have a very different chance of success. So the point here is there's a set of common underlying assumptions, but how you choose to design the study, how you choose to, the end point, the right endpoint to look at, the duration of the study, they all have a marked effect on the likelihood of success. And just look at the variation in that probability of success by looking at that heat map. Okay, I'm going to try and stop sharing and go back. Oh, so I just that case study. I mean, we could look at all sorts of different options as well. Looking at recruitment rates and some of the operational performance of the clinical trials too. But I thought it was worth just spending a moment on why do we need simulation and Keras Cloud in this context, because it is possible to go out to the literature and find estimates for probability of success or probability of technical and regulatory success from the literature. But the truth is, those are historical averages covering a wide range of different scenarios and approaches. In my view, they don't provide the kind of the relevant detail or nuance, and I think they're unlikely to apply to scenarios that might depart from convention. And that's where we need technology and data to properly evaluate the impact of doing things differently at a study, program, and portfolio level. Simulation is a really powerful tool for that kind of bottom-up, forward-looking evaluation, and it allows companies to really answer the question, which is what if we want to be different? What if we want to do things that are different to others? Simulation allows us to look at very large numbers of options all at once, and it is grounded in reality, because we can have those common sets of assumptions, and we can go back and replicate some of those historical values too. So that really validates the approach.
Thank you very much, Aiden. So kind of bringing this all back together, as we've said, we would want to create a range of options for a specific molecule or a number of assets, and then think about the places in the market where they could go. So in terms of the indications, but also in terms of the journey of getting there, going from A to B, meaning actually in the market. As Aiden illustrated, thinking about the clinical approach in different ways, inputting different assumptions and parameters. The same applies, as Alec mentioned, for also the commercial and regulatory pathways. In terms of then the path to market strategy and design, this is about, you could say, bringing it all together in these decision trees. And you see here two different ones on a slide. So this was from a case study example, not in Alzheimer's that we've just talked about, but in you could say a strategic plan to develop multiple respiratory programs, and on the other side, where there would be a prioritization for orphan indications. And what it then allows to do, if you've been exploring these different indications, is to also map out these decision trees, highlighting, you could say, the inflection points in terms of catalyst events in the market or with your program specifically, where teams and leadership need to make strategic decisions. What it also allows to do is to map out what is our time frame for further evidence generation, because as Aiden and Alex have mentioned, we will be looking at, you could say, what are the underpinning assumptions, and also that will help us to identify what are our known unknowns. Going back then to our plan, to our asymmetric learning paradigm, developing a plan to learn and thinking about the experiments you need to run in order to make better decisions is something that is a really key output of our approach. So in terms of, you could say, the outputs that going through a process like this will deliver to your organization is, on one side, you could say, a systematic approach of looking at opportunity and also really bringing in the cross-functional expertise and experience from the team, but also equally getting transparent about what are the underlying biases potentially or assumptions, and also gaps in terms of understanding where we could develop our portfolio. It also means that by keeping options alive, there's no narrowing down by anyone in the team, but really we can present these options and these analytical profiles for decision making, so that the leadership team can decide on a diversified portfolio. And then, as we mentioned, what we would also be looking into is obviously beyond, you could say, what might be ideal from a lead or launch indication perspective. How could you then quickly maximize the potential and the value of the asset by having multiple other paths to market for different indications? So bringing it all back in terms of that portfolio level decision making, our approach and what we see really helps to, you could say, take path to market strategy forward, is to explore your portfolio of options, thinking about where each asset can go and what the roadmap would be of getting there from a clinical, regulatory, and commercial point of view, obviously. Understanding the level of assumptions and the uncertainty that sits within each assumption, so that you have time to also generate evidence and run more experiments. And then to start executing those experiments, learn fast. As we've mentioned, it's about, you could say, generating insights and answering what ifs quickly. That will help you then further strengthen the options that you have in the mix, as well as then, you could say, the decisions that you ultimately take for taking them forward. And this is where, on the right-hand side, some companies, depending on their strategic approach, they might want to prioritize options that have a higher chance of commercial success, or options in the portfolio in a specific therapy area that are, you could say, lower cost to bring to market or have a lower regulatory risk. This is where every company is unique, and obviously clarity around the strategic priorities is really critical. So with that, then, thank you all very much for attending today's webinar. We'll also share the recording as well as a short output from today's meeting, and do reach out in case of any questions or further discussions desired. Thank you so much for attending.